Hello, anatomists and physiologists. This is one of the videos of our cardiovascular system series. This is part 2. This is about cardiohistology, so the tissues, the cellular level, and then we're going to start to talk about a little bit of physiology. So let's get going. Okay, so where we left off last time was talking about the pressure system of the heart. So how the heart works is just pressure differences and the physiology that we'll get to. But why the blood flows through the heart is just because there's pressure differences between the atria and the ventricles, between the ventricles and those great vessels like the aorta, like the pulmonary trunk. Those pressure differences open the valves, right? And so I always remember that blood is always going to flow to the area of low pressure. So wherever the pressure is lower, that's where, the blood will want to go, as long as the valve will allow it. So I'm going to write that in my notes here too. As long as the valve allows. Valve allows. Because we know that our AV valves only open atria to ventricles, not the opposite way, for example. And so we know that big old backflow thing prevented by valves. Even though the right side of the heart and the left side of the heart do very different jobs, pulmonary on the right and systemic circulation there on the left, we're still going to work simultaneously. We're still going to pump the same amount of blood overall. So your textbook calls the heart a double pump for this reason, because it does two jobs, but it's really just one job of circulating the blood through the body. So pressure differences, that's where we are. So speaking of pressure differences, how does blood move through the heart? So this is figure 18.7 in your text, talking about the AV valves and how they open and how they shut. So I'm going to zoom in on our first panel here. So the first panel is when the AV valves open. When the AV valves are open, that means that there's pressure differences, because when valves open, that means a pressure gradient has been established. So we know that the pressure in the atria is greater than the pressure in the ventricles. So those valves are forced to open because that blood has to go somewhere. Once the atria contract, because remember there's muscle in those walls, when the atria contract, that further pushes blood and further creates more and more pressure in this chamber to push blood into the ventricles. And this is what that valve looks like when they're open. So remember, it's like doors. And when the doors are shut, that's because a pressure difference has been established yet again. So panel A, what we just talked about there on the top, when our AV valves are open, is because the atria have a higher pressure than the ventricles. But when the opposite is true, when the ventricles have higher pressure than the atria, then the AV valves close, because the AV valves, they only operate one way. They only open down. They do not open back up. And so when the AV valves are closed, we know that the pressure in the ventricles is greater than the pressure in the atria. So this is what a closed valve looks like. So it smacks shut together. And notice that we can see those chordae tendineae, those heartstrings. That papillary muscle is holding it back. We compared the papillary muscle to a kite, a string on a kite. So if you pull that string on a kite, those chordae tendineae, that backflow is then prevented. That's super cool. Okay, so that's atria to ventricles. But what about ventricles to elsewheres? That's the next panel. This is still in your text. Oops. Still in your text here. From the ventricles, though, once the ventricles have more pressure than the next vessel, the pulmonary trunk or the aorta, that's when our semilunar valves open. Those semilunar valves open when the ventricular pressure is greater than the pulmonary trunk, than the aorta. So that's when our semilunar valves open. Our semilunar valves close when the opposite is true. As those ventricles relax, there's now less pressure. As the ventricles relax, there's less pressure. And so that's when the semilunar valves, they start to close. They smack shut, and they actually make a noise when they kind of smack shut, and the blood flow does all this stuff. So the semilunar valves close because that pressure difference is such that the ventricles have less pressure than the following vessels. Remember, our valves don't open backwards. They only open forwards. We're going to circle back to this in future videos, so to keep it in your brain. Okay, so back out here. Speaking of circulation, the heart has its own circulation. So the cardiac myocytes and all of the other things going on in the heart, they need their own blood supply, their own nutrient supply, removal of waste, etc. So in your study guide are a list of the vessels that I want you to know of the coronary circulation. Some examples of these are, let's see, the circumflex artery. I think of circumflex like as in circumnavigating the heart, like going around it. Let's see, the anterior interventricular artery, for example, goes in between the ventricles on the front. So that makes nice and good sense. But check your study guide for all of the vessels that I want you to know. Similarly, we have the veins of coronary circulation. And remember that the coronary sinus is one of those vessels that drains into the right atrium. So sinus is just a big vessel. So coronary sinus is a big heart vessel going back to the right atrium. Okay. If you go back to our lab doodles from, I believe, last class, we drew the layers of the heart wall. So I want you to pause this video, go back to those lab doodles, and put your drawing here. Layers of the heart wall, so you have it in your lecture notes too. Okay, so pause this video and come back. Welcome back. Now that you're back, we're gonna zoom in on that layer of the heart, the myocardium. That's pretty much what we're going to be talking about for the rest of this series of videos, is the myocardium and what goes on there. But I want you to think back to Bio 430, think back to when you were learning about tissues, and think back to when you were learning about cardiac muscle tissue. Cardiac muscle tissue is a lot like skeletal muscles, which we spent a lot of time in Bio 430 learning about. Cardiac muscle is involuntary, unlike skeletal muscle, so they can't control its contractions. Cardiac muscle is striated, though, so what we've seen before are sarcomeres. Look at, there's a sarcomere, there's a Z disc, an I band, an A band. Actins and myosins pulling on each other during contraction. That's great. We know about that already. I want you to have this in your brain as we go through. We have the good old sarcoplasmic reticulum, calcium storage. Calcium is such a big deal, we know, in muscle contraction. It's still a big deal in cardiac muscle contraction. But the thing about cardiac muscle cells is that instead of being long and tubular and kind of like hot dog shaped, like skeletal muscles, cardiac muscle cells are wider. They're branched. And they tend to have fewer nuclei, 1 to 2 maybe. And cardiac muscle cells are connected by special junctions called intercalated discs. So intercalated discs consist of 2 things. They consist, I'll highlight them in yellow, they consist of gap junctions. Gap junctions are holes in the membrane. So they're little pores where substances can pass through. They're little pores. This allows for rapid communication. We also have desmosomes. Desmosomes I always think of as like cellular Velcro. We're really sticking these cells together. We're really sticking those cells together. So an intercalated disc is both a gap junction and it's a desmosome. That's what's holding those cardiac muscle cells together and allowing them to communicate so rapidly. We know already from Bio 430 that muscles contract because of electrical activity. That electrical activity we learned about in Bio 430 was action potential. It's no different here, not really. So think back to Bio 430. We talked about membrane potential. Membrane potential is the charge of a cell. It's the difference in charges across the membrane, but think of it as the charge of a cell. Membrane potential is the charge. And action potential is a special kind of membrane potential. It's a rapid change in membrane potential, and it makes changes happen inside of the cell. Action potentials make actions happen inside the cell. And they consist of some things, right? And we'll talk about action potentials and reminding ourselves what happened from Bio 430. But this electrical activity, this action potential stuff, happens because of those really special properties that we have in cardiac myocytes, cardiac muscle cells. And there's two types of cardiac muscle cells. The first type are contractile cardiac muscle cells. Those are just like the regular kinds. So contractile cells, those are the regular actins and myosins pull on each other, the whole heart contracts, great, awesome. But the other type of cell are pacemaker cells, and this is the one I meant to highlight. Pacemaker cells don't contract, but they're special because they spontaneously depolarize. Depolarize is a word we haven't heard in a hot minute, so I'm going to highlight that one and define it for you here. So depolarize is when the cell gets more positive. And that's the first step of action potential, is depolarization. So that leads to action potential, AP. The cell gets more positive in depolarization, that leads to action potentials, that leads to actions happening inside the cell, and that action happening inside of the cell is muscle contraction, a heartbeat. And pacemaker cells are so super cool because pacemaker cells spontaneously depolarize, which means they don't need anybody to tell them to depolarize. They just do it on their own. This is what your textbook calls autorhythmic, or the heart has autorhythmicity because of pacemaker cells. They establish the pace, the beat of the heart. It's layered, of course, and we're going to talk about it. Okay, so that's all I wanted to talk about with this video. We're going to come back and talk even more about this stuff. Okay, I'll see you next time.